Hello students, today we are going to learn about genetic code. What is mean by genetic code? Actually we are decoding whatever the message is there in the genes. Now how it has done? In earlier structure of DNA identifying, it was very challenging work. Like that, coding the gene is again more challenging, and for that various scientists has worked a lot. The scientists involved in various disciplines like physicists, biochemists, organic chemists, geneticists has worked. It was George Gambio, that is this scientist, proposed that genetic codon is of a triplet made up of three codons. Now, how he has proposed that the codon is made up of a triplet, means three successive nucleotides will give a one codon. So, for that see, we know that in our body there are 20 different types of amino acids, and to code that 20 different amino acids we have how many types of nitrogen base? Only 4 types, that is A, T, G, C. See his calculation, what he has decided. If the 4 codons are there, and in that if we will take combination of only one codon for one amino acid, means if A codes for one amino acid, T codes for second amino acid, C code for third, and G code for fourth. So in this 4 codons are there, it will code for only 4 proteins. So is it possible to code 20 amino acids by only 4 codons? So this is what one base code, that is 4 raised to 1, gives only 4 combination, means only 4 amino acids will be coded. Now the second criteria, he said that if we take the combination of 2 bases, that is AT, GC, GA, like that, then it will give 4 into 4. Then total 60 different types of combinations we will get. So means that these 16 different combinations are there. This will represent 16 different amino acids. And how many amino acids we have? 20 amino acids. So is it sufficient, these 16 combinations for 20 amino acids? No, it's not sufficient. So he decided that the 3 base codes, that is 4 into 4 into 4. So total we got 64 types of combination. So these 64 types of combinations are more than enough to code only 20 types of amino acids. So here the George Gambio has given that the codon is of 3 base code. Now 3 base code means any 3 successive nucleotides when comes together, it will code for any one type of amino acids. So they are more enough than coding 20 types of amino acids. After the George, Marshall and Nehanberg, these are the 2 scientists who have worked a lot to find the codons for different types of amino acids. Now what they have done, they have taken the codon, only one type of codon, that is that is uracil, nucleotides of uracil they have taken. Then this created artificially poly-U, that is artificial poly-U mRNA is formed, and along with that mRNA they added ribosomes, tRNA and different types of amino acids in a test tube. Now all these components are required to form a protein. So as they have only uracil type of nitrogen base, then at the end of this they got only one type of amino acids chain, that is phenylalanine. Everywhere they got the phenylalanine. Means 3 successive uracils will code for phenylalanine amino acid. So in this way they have done different experiments in a various combinations, and they have find out that which types of codons are there, they will code for which amino acids. For that, one table is also given in your textbook. Now in this you can see that here triplets are given. Now how these triplets are there? See here UCAG, UCAG, and here also UCAG, it is given for every column. So in this every triplet, all of you have learned the Mendelian dihybrid cross, monohybrid cross, and in that Punnett square also you have learned. So you know that in the Punnett square how we are taking the gametes, one from male gamete and another from the female gamete, right? So the same here it will be done. This will be the first letter U, then second letter from here, and the third letter from here. So this box, the first U, and this U, then C, then A, then G. So we got here 4 triplet codon. Like that you can do anywhere, like this A first, then here also A, and from here U. Like this G, then from here C, and from here U. So in this, if you will see, everywhere first 2 codons are there. It is same in this box. Then here also the first 2 are same. Here also the first 2 are same. Only the last codon is changed, and because of that sometimes all these 4 types of codons representing only one type of amino acid. Like here leucine is there, proline is there, serine is there. So in this way, most important is the first and second codon. So the last codon may be any of these 4 will represent for leucine only. Last codon here also anyone will be there out of the 4, and if the first and second is the CC, then it will be a proline. So in this way 20 different amino acids are given here with their triplet codons. If you will see this table here, UAA, UAG and UGA, 3 codons are there which is given in bold, and ahead it is given no any amino acid name. It is given as a stop. So these 3 codons are there. They did not code for any amino acids. So that's why they are known as the stop codon or nonsense codon. So whenever in the mRNA sequence, whenever these 3 sequence will come, there the process of adding amino acids will be stopped. Means translation process will be stopped. Like that, if you will see here, AUG, it will code for methionine. Then here this codon is also act as a starter codon. Means whenever mRNA is translated, that time this will be the first codon which will code for the met, and this will be the first amino acid for every protein formed. Right. So this is the starter and this will be a stop codon. So AUG plays dual function. It will be started as well as it also code for met. While these 3 are there, they will not code for any amino acid. So out of 64 combinations, we have only 61 codons which will be code for 20 amino acids, and these 3 will not code for any amino acid. Here are some of the silent features of the genetic code, that the genetic codes are universal. Universal means all the organisms will show the same types of codons and they will code for the same amino acid. As you know, earlier U. If this triplet is there, this will be code for phenylalanine. This is in human beings, right? But if you will check this in even bacteria, then it will code for phenylalanine only in bacteria. Means any living organism if you will take and in that U codon is there, then it will be code for phenylalanine only. So this means the genetic code is a universal. Codons code for any amino acid in any organism, be it be a bacterium or a human being. Second is that second silent feature is non-ambiguous. The genetic code is non-ambiguous. Non-ambiguous means what? Only one codon codes for one amino acid. Every codon is there. We have seen that there 61 combinations are there. In that, any codon does not code for two amino acids. For example, again triple U, that is U codes for phenylalanine. Then this U code will not code for any other amino acid, right? So this means that non-ambiguous. Each codon code for only one amino acid. So the genetic code is unambiguous and specific. This is also called as unambiguous or specific for that particular amino acid. Next silent feature is the commaless. Genetic code is a commaless. Commaless means when we write something in the language, we are giving full stop, comma, any other punctuations. But in case of genetics, it is simply start from the first, then it will be a continue. There is no comma, no full stop, no any other punctuation. So it is a continuous code. So that's why it is called as a commaless. The codons are read in 5' to 3' direction and no punctuations are there. Next silent feature is degenerate. Genetic code is a degenerate. Means some amino acids are coded by more than one codon. The genetic code is said to be degenerate. For that, just see once again here. Means U also code for phenylalanine and UUC also codes for phenylalanine. Or CUU or CUA. Any of these four types of codons all code for leucine. Or again you see CAU or CAC codes for histine, right? So in this way, more than one type of codons code for the same type of amino acid. Means leucine is coded by 1, 2, 3, 4 types of amino acids. So in this way, if we will see, then this is what genetic code is a degenerate. Means more than one codons code for only one type of amino acid. Next silent feature is non-overlapping. 3 successive nitrogen base code for only one amino acid and they will not overlap. Means here if you will say UAA, the next codon will be start after this, that is UAG. It will not be AAU or it will not be AUA. So overlapping is not there. So that's why the next silent feature is non-overlapping genetic code. Then nonsense codon. Nonsense codon, I said earlier that UAA, UAG and UGA. These are the 3 codons which does not code for any amino acids. That's why their termination of the translation process takes place, and that's why they are called as a nonsense codon or stop codon. Next is that the genetic code is a linear, means in only one particular chain only and continuous one from 3' to 5' direction. Sequence of amino acids present in polypeptide chain corresponds to the sequence of nitrogen base of DNA with 3 successive nitrogen base forming a single codon. This means a linear. As Gambiu said that the codon is of triplet, means 3 successive nitrogen base pairs are there that will form a particular triplet, and there are 64 combinations of codons are there. out of that 61 codons code for 20 amino acids and 3 will be non-coding, stop codons are there. And as I said earlier, AUG is a codon, is a initiating codon, and it also codes for a MET. So it plays a dual function. So these are some of the silent features of the genetic code which is given in your textbook. Now based on this, 2 or 3 marks question will be asked for the silent features of the genetic code. 6 features you have to write which is given in your textbook. Next thing is that now you will ask me that this table do we need to prepare for exam? Majoritarily in this, the MET codon you have to remember, and these 3 stop codons are there that need to be remembered from the exam point of view. Rest one, if any particular codons are there, if possible, then in a group you can remember like this. This is a very easy way to make a box, that is UCAG. This is the same sequence we will found in all 3 sides, and only as like a Mendel's Punnett square you have to take from 3 sides and make the codon. So you can remember in this way that this 4 for valine, this 4 for alanine, this 4 for thrombin, this 4 for proline. In this way you can remember few of the codons wherever it is possible. But starting codon and stop codons need to be remembered for exam, right? Another thing is that the genetic code, how it is invented, it is triplet only, and this is invented by whom? So this type 1 or 2 marks question will be based on that discovery of genetic code, based on this, and rest one all are easy, you can prepare well, and best of luck.